Hi, my name is Nicholas Kaji and this is the Molecular Biology Seminary. On this video, I'll be talking to you about the microRNA-323, the docetaxel resistance, and also the repression of p73 protein. Before we start, we have to know a few concepts to get along with this topic. Okay, so let's start with cancer. Cancer is the abnormal growth of cells that can lead to different multiple, local, and systemic problems. But first, we have to know that in this study, they used the prostate cancer cells. Prostate cancer is commonly caused in males because males are the ones that have the prostate. The prostate is a gland that can produce a bunch amount of seminal fluid. It's quite important for the semen. So the prostate cancer is commonly caused by adenocarcinomas and is one of the principal mortal causes in men. Okay, so we also have to talk about the p73 protein, which is quite similar to the p53 protein. This protein is the one that regulates the cell cycle, so they can induce apoptotic procedure. We also have to talk about the docetaxel. The docetaxel is a chemotherapeutic agent that they use on these prostate cancer cells. Now, the microRNA is the small non-coding RNA that negatively regulate gene expression. But on this experiment, they used the microRNA-323, which is an specific microRNA that plays an important role in prostate cancer cell proliferation. The general objective was to determine the function of microRNA-323 on the prostate cancer cell growth and what it does to the cell, to p73 protein, and also the relation with docetaxel resistance. Methods they use for the culture: ideal medium, ideal temperature, and the ideal time. The PCR-RTQ. The PCR is a technique used to amplify a specific segment of DNA. They use the reverse transcriptase because they took a segment of RNA, and the Q represents the quantification process. They use the plasmid as the vector to transport the information, but the method they used was the transfection, which they exactly use to create vector transfected cells and microRNA-323 overexpression cells. But they have to show these results by a reporter, and the reporter they used was a luciferase, which is a bioluminescence protein. The other method they used was the MTT, which is a cytotoxic assay used to watch cell damage to certain treatment, in this case to docetaxel, because they had to prove it wasn't too cytotoxic for the cell. The rats, they used the rats because was the in vivo model. Because they were studying apoptosis, they needed an apoptotic analysis, which is a method used to prove what was happening with the cancer cell apoptosis due to the given treatment. They also used the Western blot, which is a protein electrophoresis method used to prove an antigen-antibody reaction, which I'm going to explain later on. Now we're going to start with the results. In the figure 1, in the graphic C, we can see the number of colonies formed by microRNA-323 overexpressing PC3 cells were higher than the vector transfected PC3 cells. On the same figure 1, but on the D graphic, we can see an in vivo tumorigenic study that proves us the other result from the culture. It gives us the information that microRNA-323 overexpressing cells had a more proliferative rate than the vector transfected ones. With the results on figure 2, we're going to describe the ones on the graphic D. But first, we have to understand what annexin 5 is, which is a protein used in the flow cytometry that has the ability to bind with phosphatidylserine, which is an apoptosis marker. So here, after the annexin 5 is stating that microRNA-323 led to an increase in the apoptosis of PC3 cells versus the control group. On this same figure 2 but on the E graphic, they used the Western blot analysis that indicated the presence of the caspase-9 and 3 on the anti-microRNA-323 PC3 cells. This proves that microRNA-323 depletion induces cell cycle arrest and apoptosis in prostate cancer cells. On the figure 3, the graphic G shows us the real-time PCR analysis and the Western blot. On the Western blot, they always use actin because it's a protein that is always present on every single membrane of the body. So, what this is showing us is that there was no significance of MDR1 protein, so microRNA-323 did not alter MDR1 efflux mechanism protein. On this figure 4 on the graphic A, we can see the mutated p73 3-UTR fragment cloned to the luciferase reporter vector and then transfected with microRNA expressing vector. In this image exactly we can see the mutant part of the strand which is affecting the p73. Now for this image C, we have two gels showing us one with the vector and one with the microRNA-323. They're showing us the expression of the p73 protein, which was higher on the vector one than on the microRNA-323. This is guiding us to the same hypothesis we had on the beginning, which is that microRNA-323 reduces the expression of p73 protein. We haven't talked a lot about the apoptosis process, but it is quite important for this article because, as I already said before, the p73 acts just like the p53, inducing the expression of the caspase-9 and caspase-3. So, here on this Western blot gel analysis, they took a vector empty and an overexpression with the p73. So, the overexpressive p73 showed the proliferation of the caspase-9 and caspase-3, which leads us to an apoptosis process. For the discussion, I chose four authors. So, Q. Gall said that the microRNA-323 inhibits the prostate cancer vascularization through a adiponectin receptor. So this was false because they never even said that on the article. The next one is C. Concoran said that the microRNA is an intracellular and exosomal predictive biomarker for response to docetaxel with clinical relevance to prostate cancer progression, and it was completely true. M. He said that the downregulation of the p73 contributes to androgen independence, and this was false because they proved that the downregulation of p73 expressed androgen dependence. This last author said that the microRNAs had a role on the docetaxel resistance, and this was true. From this article, we can conclude that the p73 is downregulated by the microRNA-323. We can also conclude that docetaxel resistance is caused by other genes different from the MDR1 gene. Cancer is the abnormal cellular proliferation. It affects both men and women. Women tends to suffer endometrial cancer, breast cancer, and also lung cancer. But we are going to focus on the men. Men tends to suffer colorectal cancer, lung cancer also, and the prostate cancer. The prostate cancer causing the overexpression of the microRNA-323 gene, leading to the docetaxel resistance and to the downregulation of the p73 protein that inhibits the apoptosis. Thank you.